Okay, our next podcast is going to go into looking at the basis of inheritance in the molecule of DNA. We can begin by saying DNA makes up our chromosomes. We looked at that when we looked at cell division and the structure of the chromosomes. And so our genes, our inheritance is carried on segments of that DNA on the chromosomes. And we'll look at the DNA in a minute, but the chromosomes we can track through meiosis. So we learned about that process in an earlier unit. And so in this slide here, you can see that through meiosis, the production of the sperm and egg, you can see the path that these chromosomes, so we're tracking these traits right here, the yellow round traits of the pea pods, and then the green wrinkled traits are the recessive traits here, and we can track those all the way down through fertilization and then into the meiosis, all the phases of meiosis until we get all of our particular alleles separated out in the gametes through that production. So we can see that through that laws of assortment, excuse me, that the alleles go through when they're dividing up in meiosis. So this kind of shows us the basis of chromosomes carrying the genetic information into our gametes. And then we can talk about these chromosomes, what is on there that carries the traits, and that is the chromosomes are made up of our DNA strands. DNA is a molecule, a long molecule that's made up of a sequence of subunits called nucleotides. And that structure was first discovered by Watson and Crick. And so they're the famous discoverers of the structure of DNA. Prior to this, we mentioned in our outline there some early studies that were shown that kind of showed that genetic information is found or in the nucleus. So that was earlier studies, the study with the Cetabularia, and then actually Griffin and Avery did a study with pathogenic bacteria that infected mice, and their studies showed that genetic information was being transferred from one cell to another. So there was some earlier studies prior to Watson and Crick about this genetic material that was in the nucleus, and then the chromosomes were discovered, and then Watson and Crick uncovered the material within. Here is the Griffin and Avery study is right here. So this shows kind of what they did. They would take these are the bacterial cells that they would inject. These are some lethal bacteria that would be injected into the mouse. The mouse would die there, and here's a non-pathogenic strain of that bacteria injected into the mouse. The mouse did not die. And then these S cells, these pathogenic cells, were heat killed, where they would no longer be alive to cause the symptoms to kill the mouse. But when these two were combined, either one of them by themselves would not kill the mouse. But when they are combined and injected into the mouse died, and then samples would be taken, and living S cells would show up there. And so what was happening here in these bacterial cells, genetic information was transferring from one cell to the next. So again, some early genetic studies showing us that some material was transferring, some genetic material was transferring in between the cells. And then, so Watson and Crick were some of the first to discover what that structure was, but they had some other studies that they used to guide them. One of those was Rosalind Franklin. Now, Rosalind Franklin, she did some different types of diffraction X-ray images of DNA strands, and that's what this is right here. So Watson and Crick had this to look at, but this doesn't tell us exactly the structure. What this is that they determined, and we now know, is that this is the sugar and the base that forms the helical shape of the DNA strand. This right here in the middle would be the bases that connect the helical shape, the helical ribbons of the sugars and the bases. So this really helped Watson and Crick discover that structure of the DNA. So now we know that that structure is basically made up of repeating subunits called nucleotides. So one nucleotide is made up of a phosphate, a sugar, deoxyribose is the sugar, a phosphate group, a sugar, and one base, nitrogenous base. All right. The sugar and the phosphate are the same in all the nucleotides that make up the long strand, but what differs is which base is going to be attached. And there are 4 different bases of DNA. There's adenine, thymine, cytosine, and guanine. So that's kind of our alphabet of genetics, A, T, C's, and G's. And this is what makes the code which will determine the particular traits. So this traits are coded by the sequence of these A, T, C's, and G's. So brown hair would have a certain sequence, and blonde hair would have a different sequence of these A, T, C's, and G's. So DNA is just made up of a bunch of these nucleotides stacked together to make a long strand, and then 2 strands actually come together, and that's why we call it a double helix, because there's actually 2 strands that hook together. So that's the basic structure. Now there's some different ways of describing the structure of DNA. Now your book will use all of these. This is sometimes called the ribbon model, where the sugar and the phosphate are just represented by these blue ribbons, and the bases of those strands are right here in the middle. All right, so the bases are, again, abbreviated A, T, C, and G for adenine, thymine, cytosine, and guanine. So if you notice here, all the way down the strand where the 2 strands hook together, C's always match up with G's and A's always match up with T's as we work our way down the strand. That's called complementary base pairing. It's what that matching is. So that's the ribbon model. This is a chemical structure model where we can actually see each part of that nucleotide, the phosphate, the sugar, and then the base. They are hooked together, so this is one end of the molecule. The molecule is unwound, so this shows it wound together in its spiral or helix. This has it unwound so you can see it better. But you have a phosphate, a sugar, and a base. And all the way down, another phosphate, sugar, and base. Now, there's a top and bottom to these strands as well. The top we can refer to here is the 5' end, and the 3' end down here. The other side is in the opposite direction, so they are turned over and go this direction. 3' end on this end and 5' down here. All right, so this is, that's called the chemical model, and then this is the space filling model, which just represents each of those parts of the molecule by these little balls there. Okay, so Watson and Crick basically built their model, this double helix, using Rosalind Franklin's X-ray and technology that they used, X-ray images. Okay, so the complementary base pairing, we can see that here. So here's our strand of DNA. DNA actually, when it copies itself or replicates, it replicates what's called semi-conservatively, meaning that it keeps part of the molecule the same and then it builds upon that. So when DNA copies itself, when it's getting ready to make more chromosomes, when cells are dividing, it will split in half or unzip. Geneticists refer to it. It'll just unzip right down the middle. And so here's our DNA strand, and it splits apart. And what comes in are new nucleotides will come in and attach to these open ends of the nucleotides on each side. And so the light blue over here are the new nucleotides that have come in. And so now we have two strands. The dark blue is from our original DNA, and our light blue is new nucleotides that come in to complete these two daughter DNA molecules. When this splitting apart takes place, it takes place at replication forks. That's the Y region where it splits open. You can see that here. So the Y looks like a Y. It's where the DNA has split apart, and so that's where this takes place. This building of these new strands as the cell divides. And enzymes are around to help with this speeding up of this process. Enzymes help with the addition of the new nucleotides to the strands. Okay, and that's where we end for this kind of introduction to the DNA, the structure that carries the genes. So the segments on there are called a gene, and that's what we're going to get into next, is looking at the genes and the functions that they carry out in the cell.